Hello everyone and welcome to Biology with Zakia Sultana. In this video we are going to discuss a topic which seems to be difficult but actually is very easy. This topic is relevant for both IGCSE and IAL biology students. Our topic for today is transcription. So let's get started. Transcription is the first stage for protein synthesis and it takes place in the nucleus of a cell. The second step, which follows transcription, is translation, which takes place in ribosome. In this video, we will only focus on transcription. Now, what is transcription? Transcription is the production of an mRNA, that is messenger RNA, from a segment of a DNA molecule. The question arises, why is transcription important at all? So, we all know that the DNA molecule contains the genetic information or the code or the recipe for making a protein, and the protein is made in ribosome. But unfortunately, this DNA molecule is so large that it cannot travel out of the nucleus through these tiny nuclear pores to go out to the ribosome and cook, synthesize, or make the protein or the polypeptide. So what's the solution? The DNA, it would send its messenger, which is the messenger RNA, out of the nucleus to the ribosome in the cytoplasm to make the protein. It is possible for mRNA to move out of the nucleus because it is small enough to be able to pass through the nuclear pores. Now, let's see the steps in transcription. The first step in transcription is to unwind and unzip the gene portion of DNA. So, we should remember that not the whole DNA would unwind, it is only the gene portion of DNA which would unwind and unzip with the help of the enzyme DNA helicase. In the second step, the nitrogenous bases in the DNA nucleotides of the unwound section of DNA are now exposed. We should also keep in mind that although DNA has a double helix structure, but not both the strands are part of the same gene, a single gene is always on a single strand. And that is why we are only focusing on one strand and not both. When the nitrogenous bases on the DNA molecule are exposed, some complementary RNA nucleotides, which are already present in the nucleoplasm, would align with the complementary exposed DNA nucleotides. These complementary pair would form hydrogen bonds according to complementary base pairing rule. And to do this, the enzyme which helps is known as RNA polymerase. So you can see uracil binds with adenine to form hydrogen bond, guanine will come and bind to cytosine, adenine with thymine, cytosine with guanine, and so on and so forth. The next step is the formation of phosphodiester bond between the RNA nucleotides. After this has been formed, the RNA, which is now known as the mRNA, would detach from the DNA and exit the nucleus through these tiny nuclear pores. This mRNA, being much smaller than the DNA, can easily pass out through these nuclear pores and carry the message to the ribosome. This ends the process of transcription. This would be followed by the process of translation in ribosome to produce a protein, or more precisely said, a polypeptide chain. So that's all for today. Thank you everyone for watching. In my next video, I would talk about translation. So keep an eye on the page and YouTube channel. Thank you and bye-bye.